In your reading assignment yesterday, you got introduced to a lot of terminology, and the terminology describes the principles that Mendel discovered, but that terminology is what we are commonly using now. And so hopefully you realize that the factors that we were talking about in the first video, we're now going to call those things alleles. And the fact that the tallness factor, or the tall allele, would overcome or would overshadow the shortness one, we say that the tallness then is dominant over the gene or the allele for short. And so we've got dominant traits and we have recessive traits. If you have a dominant and a recessive trait both coded for in the same organism, the dominant trait is the one that's going to show up, even though the allele or the gene or the factor for the shortness trait, for the recessive trait, is still going to be present. We also talk about whether a organism is homozygous for a trait or whether it's heterozygous for a trait. Now, homo and hetero are two prefixes that probably in today's society everybody's pretty familiar with what they mean. If you have an organism that is homozygous, then that means both of its alleles for the trait are exactly the same. If you have an organism that is heterozygous, then that means both of the alleles for the trait are going to be different. So what we want to do is we want to start looking at some Punnett squares to see how we can predict what traits the offspring could potentially have given the characteristics of the parents. So I have an example written up here on the board. A pea plant with wrinkled peas, which is a recessive trait, is crossed with a heterozygous plant. So recall again that the texture for the peas, and this was in the very first section, to give you a whole list of different characteristics that pea plants could have, and it told you, well, this is dominant and this is recessive. And whether the peas were wrinkled or whether they were smooth was one of the characteristics. So we're going to be looking at pea plants, a pea plant with wrinkled peas. Now, since this is the recessive trait, what that means is that the parent has to have both alleles the same if this plant is going to show that it has wrinkled peas. Now, you can pick any letter that you like, and it doesn't matter what letters you pick. We're just going to use capital letters and small letters. The one thing I want to caution you about is that whatever letter you pick, you need to be able to tell the difference between your capital letters and your little letters. So if you're one of these people that your handwriting is kind of sloppy and you're not always super careful about how tall your letters are, then you need to be extra careful to make sure that you choose letters where the capital and the small letter are very different. If your handwriting is really, really neat and you're not going to have any trouble telling the difference between a capital S and a little s, then it doesn't matter, okay? But you need to be able to tell the difference. Now, for this particular example, I just chose I'm going to use Q. Doesn't matter what letter you use as long as you can tell the difference. So, for wrinkled peas, this genotype, which is what it's called, is going to have both recessive traits. I'm going to spread that out a little bit. Both recessive traits. So this guy is our wrinkled one here because he has two recessive traits. Okay, now, this plant is then going to be crossed with a heterozygous plant. This one is homozygous because both of these are the same, and the only way that you can show a recessive trait is if both of the alleles are the same. In a heterozygous plant, that means that I'm going to have one for the dominant trait and one for the recessive trait. So that means this plant over here is actually going to have peas that are smooth in the way that it actually looks, which is called its phenotype. So what happens when I cross these two together? Well, I'm going to draw my Punnett square like this, and then I'm going to cross them together. So my first offspring will have these two traits in it, so I'll have a capital Q and a little q, and that's what this one will have also. This one will have a capital Q and a little q. Down on the bottom, both of these are going to have little q's, and this one in the bottom right-hand square, this will also have two little q's. So what do my plants look like? Well, I can analyze this data from two different perspectives. I can look at it in terms of the genotype, and I can look at it in terms of the phenotype. Now, the genotype means specifically what alleles do they have. These are the alleles, so what alleles are actually present. Well, in 2 of the allele, or in 2 of the offspring, I have a capital Q and a little q. And in fact, in 4 of the possible offspring, 2 of the 4 of them have a capital Q and a little q. 2 of the 4 also have a pair of little q's. So again, this is 2 out of 4. Now, hopefully you remember from your math class how to take a fraction and convert it to a percentage. If you don't remember, I'll do a real quick review for you. And I forgot to mention too, it's perfectly okay if you want to use a calculator for this particular module. This is not a math test. This is a test to make sure you understand how to figure out what kind of offspring, what percentages are going to be. So if you want to use the calculator, I'm fine with that. These hopefully would be pretty simple. 2 out of 4, if you're going to calculate this, you would take 2, you divide it by 4, and then if you're going to turn it into a percentage, you have to multiply it by 100. So when you do that, 2 divided by 4 times 100 is going to give me a percentage of 50%, okay, which is half. And then this is also going to be 2 out of 4, or 1/2, which is also 50%. So those are my genotypes. The genotypes are going to be big Q, little q, and then 2 little q's. Now, what are the phenotypes? Because in some instances, we might almost classify the phenotypes as being more important because that's what the plants are actually going to look like. Well, what are these plants going to look like? Are they going to have smooth peas, or are they going to have wrinkled peas? Well, this allele is going to overpower this one. This one is dominant. And so the dominant trait is the one that's going to show up. So what that means is that for smooth peas, because that's what these are going to show up as, 2 of the 4 are going to be smooth, which is 50%. But the ones that have little q, those are going to show up with wrinkled peas, which is also 2 out of 4. Or 50%. Okay, so these are the genotypes. That's what the genes, that's what the actual alleles look like. But these are the phenotypes. What does it look like in actual practice? I can also take these and put them into a ratio. Since I have basically one of these for every one of these that I have, my ratio here is a 1-to-1 ratio. It comes up exactly even. I have one of these for one of these. One of these for one of these, okay? And so the phenotype is actually also going to have the same ratio. It's also going to be a 1-to-1 ratio. For every smooth plant, I also have a wrinkled pea plant. For this smooth one, I also have a wrinkled one. So it's a 1-to-1 ratio. Now, let's look at one that is a little bit more complex and make sure that you understand how to do that one. Because sometimes you may have some dominant traits that are going to show up, but the genotype may not always be the same. All right, the example I'm actually going to do is example 7.2 in your book, and I want to make sure that you understand, again, where these genotypes and phenotypes are coming from. So example 7.2 says a heterozygous purple-flowered pea plant is crossed with a heterozygous purple-flowered pea plant. Okay, so first of all, heterozygous, what's that mean? Heterozygous means that the 2 alleles do not match with each other. Okay, we also know that if it's heterozygous, the dominant allele is the one that's going to show up, and so if it's heterozygous and it's purple flower, that must mean that the purple flowers are going to be the dominant ones. Okay, so if I have a heterozygous purple-flowered pea plant, and in this particular example, they used F's. You could use P's. You could use whatever letter you want. So if it's heterozygous, a capital F is going to mean that it's got purple flowers. A little f would mean that it has white flowers, because in pea plants, that's what's dominant. So a heterozygous purple-flowered pea plant is crossed with another heterozygous purple flowered pea plant. So that means that this one is going to have exactly the same alleles as this parent does. So we're going to draw our Punnett square again. And again, remember that because you've got these diploid cells that through meiosis are going to go down and become haploid cells, only one of the traits of the parent is going to be possible for each of the offspring. So that's why we're dividing it up this way. So never lose sight of the fact that that's where all this is coming from. So if I look at this one, I'm going to get a capital F from here and a capital F from here. So I have two capital Fs. This one is going to be a capital F and a little f. This one is also going to be a capital F and a little f. And remember, I always write the capital letter first, no matter which parent it comes from, because that's really immaterial. And then the last category here, I'm going to have a little f and a little f. Okay, now let's do the same thing that we did before, and let's analyze both the genotypes and the phenotypes. So the genotypes, it looks like I actually have three different possibilities here. I have a capital F, and only one out of my 4 offspring has that particular genotype. So that is a 1 out of 4. I have 2 other offspring who both have a capital F and a little f. And so since there are 2 out of the 4 there, that would be the fractional, the ratio there. And then I have 1 offspring that's going to have 2 little f's, so that's going to be 1 out of 4. So if I then take these guys and I turn them into percentages, it's going to be 25% this one, it's going to be 50% for this one, and 25% for this one. Now, when I do this, I normally do start out with a homozygous dominant, then a heterozygous, and then a homozygous recessive. I usually do it in this order so that when I give you the ratio, people are going to know what we're talking about. So since I have 1 to 2 to 1, that's what the ratio is going to be. 1 to 2 to 1. So here's the 1, here's the 2, here's the 1. Okay, now those are the genotypes, but what do the plants actually look like? What they look like are the phenotypes. Now here, it's not going to match like it did the other time, because if you notice this one and this one, this is going to have what color flowers? Hopefully you said purple, because both of these have a capital F. What color flowers is this going to have? Yeah, this is going to have purple flowers also. So both of these guys are going to have purple flowers, but this one is going to have what color? Yeah, this is going to have white flowers. So I'm going to have 3 purple flowered pea plants, and I'm going to have 1 white flowered pea plant. So the fraction, there were still 4 possibilities, so this is 3 out of 4, which is 75%. This is going to be 1 out of 4, which is going to be 25%. So the ratio for the genotypes is actually going to be 3 to 1. So here's your 3 and there's your 1. That's where that ratio comes from. So even though the ratios are different, the ratios are going to be different because you've got 1 homozygous and then you've got 2 that are heterozygous, but they both show up with the same phenotype down here in the bottom. Okay, you guys are going to get some practice doing these, and then when we come back in the next video, we're going to look at how this really plays out from one generation to the next.